I'm Sherry Leese. I'm the coordinator, the program leader for Great Plains Fire Science Exchange, and I'll be moderating the presentation today. A quick note about the exchange: we are dedicated to making fire science accessible in the region, and our focus is on grassland systems of the Great Plains. And as you're coming in, I think a lot of you have already seen the poll that I put out. We just wanted to get an idea of who's joining us today. So I'll get that out of the way here in a second after my introduction. I did want to mention that this presentation is sponsored by eExtension. That's an online extension website where you can interface with professionals in the field if you have environmental questions. And so we are working with those folks. And the Joint Fire Science Program funds us as part of a national network to communicate fire science. We do have an additional webinar scheduled in January, and that's going to be looking at the effects of fire on hornflies and ticks and the effects on livestock production in January. You can also find us next week at the National Grazing Lands meeting in Grapevine, Texas, and then also at the Society for Range Management meeting in Corpus Christi, Texas in end of January, early February. So let's see. All right. So I'm excited to introduce our speaker for today. Estelle Levitin is faculty at the University of Tulsa with a specialization in mycology and botany. She's been working on pollen forecasting in the aerobiology lab at the University of Tulsa. And it's a new word for me. It's something new I've learned today. Aerobiology is the study of movement of biological particles within the atmosphere or indoor environments as well. And I want to thank you for being with us today, Dr. Levitin.
Oh,
And
you're welcome.
just so participants will be muted, and you can type your questions into the chat box, and we'll take those at the end. So go ahead, Estelle.
Okay, thank you. I hope you can hear me.
I can.
Oh, good. Well, I'm happy to talk to you about one aerobiology and one of my favorite pollen types, and that's juniperus. I say favorite in a very lighthearted way. I've been studying juniperus pollen for over 30 years, and it's really a major allergy problem in many parts of the world. But I thought I would start off talking about some background about allergens in general, and especially respiratory allergies, and then talk a little bit about my sampling equipment and a little bit about pollen seasons in general, and then the rest of the time we'll spend on juniperus pollen and pollen transport and some of the work I've been doing in the lab here. So about 25% of the population has allergies, and allergic rhinitis and allergic asthma are the most common forms of allergic diseases. Allergic rhinitis is what we commonly call hay fever. And individuals with allergic disease are genetically predisposed to produce a certain type of antibody, it's an IgE antibody, against common environmental substances. We normally think of our immune system reacting to pathogens, but in the case of these IgE antibodies, they're triggered by common environmental molecules or particles. I indicated that about 25% of the population has allergies, but a recent epidemiological study showed that 54.6% of the population tested was positive for one or more environmental allergens, which suggests that the 25% estimate may actually be low. And allergies are triggered by environmental allergens, both indoors or outdoors, and pollen is a major trigger of both allergic rhinitis and asthma. So a little bit more about hay fever and asthma. In the U.S., the estimates are about 50 million people are afflicted, and globally, of course, it's much larger, and we know that all types of allergies are increasing globally. The typical symptoms of hay fever include sneezing, runny nose, nasal congestion, watery eyes, itchy ears. And I will say that some of the figures about the estimates, the 50 million people in the U.S., many people consider that low because many people self-medicate their allergies with many of the over-the-counter antihistamines and nasal sprays. So we don't have a solid figure for the number affected with allergic rhinitis. Allergic rhinitis can continue on into more serious conditions. Sinusitis is common, ear infections are common, nasal polyps not as common, and asthma can also develop from severe allergic reactions. Health expenditures for treating hay fever are estimated at about $13.5 billion, and that figure is about 5 years old. And this includes visits to doctors and prescription medication. And the figures do not include over-the-counter medication. So this is a visual of the symptoms, and many of us can identify with that. I have allergies myself, and I can easily identify with everything there. Asthma is a more serious condition. It is a chronic breathing disorder that's characterized by three things: airway inflammation, bronchial constriction, and excessive mucus secretion. And the three are usually happening concurrently and result in wheezing, coughing, and choking. Asthma outbreaks, asthma episodes are responsible for anywhere from 3,500 to 5,000 deaths per year in the United States. And asthma attacks can be triggered by exposure to allergens, but also can be triggered by infections, stress, exercise, temperature change. Physicians consider asthma more of a syndrome rather than a single disease, since it has multiple triggers in addition to the allergens. However, the majority of people with asthma are allergic to one or more environmental allergens, and the percentages vary with the study. In one study, 85% of the asthmatics showed to have allergic asthma, but that was only in one study. Most of the other studies show that about 50% are allergic to an environmental trigger. And this visual depicts what's happening on, let me see if I can get this. Here on this side, you have normal bronchial tubes, normal airways. But here you have a typical inflamed bronchial tube, the inflammation making the walls of the bronchial tubes expand, the muscles around the bronchial tubes constrict, and mucus secretion, and all of which are narrowing the airways, making breathing difficult. Asthma has been increasing, and in the U.S. today, it's estimated that about 25.5 million people suffer from asthma, and this is based on 2012 data. The number of people with a lifetime diagnosis of asthma is close to 40 million. The current breakdown includes about 18.7 million adults, and that's 8% of the adult population, and 6.8 million children. That's over 9% of the childhood population. And we know that there's over close to 20 billion in direct medical cost, plus another 36 billion in lost productivity. And since 1980, asthma prevalence has been increasing, and especially in children, although the increase has been leveling off in the past few years. It's still increasing, but the rate has leveled off. And we tend to see the increase, especially in inner cities, and especially among children in inner cities. So we're going on the assumption that pollen can trigger both of those allergic diseases, both the allergic rhinitis or hay fever and asthma. So let's switch over and talk about the plants. And we typically call these hay fever plants. And hay fever plants have the ability to produce abundant quantities of lightweight pollen that's easily dispersed by wind. The pollen can remain airborne for extended periods of time and often be carried long distances. And I'll be talking about this later on. The majority settle out close to their source, and a lot of the older literature says that most important hay fever plants grow close to human populations. And I just added or not, and we can revisit that at the end of the talk. And the actual component in the pollen are either proteins or glycoproteins that are the actual allergenic molecules. And these molecules are released from the pollen grain in our nasal passages and picked up by our tissues. And this is what's triggering the allergic response. Typically, flowers among the angiosperms, among the flowering plants that produce allergenic pollen are wind-pollinated plants. And the flowers in these plants are typically small, they're inconspicuous, they lack petals, they lack sepals, they are evolutionary programmed to produce large quantities of pollen. And the ones illustrated here, this is elm. These are the flowers on elm, ragweed flowers. These are oak flowers, and you can see in many of these the flowers are produced in an inflorescence rather than solitary flowers. These are grass flowers. And here is cedar or juniperus, and these are the pollen cones in juniperus. And generally, these are in size maybe 5 to 8 millimeters, so less than a centimeter in size. These are very small cones. I've been using a Burkhardt air sampler at the University of Tulsa for 29 years, almost to the day. Before that, I was using a Rotarod sampler, but I'm not going to discuss any of the Rotarod data since they are not quite comparable in lumping them together. And these are volumetric samples. They take in air through an intake orifice, and the intake orifice is right there. The wind vane here allows the head to rotate freely so that the intake orifice is always facing into the wind. There's a pump here at the base that draws in air at 10 liters a minute, which is pretty similar to a breathing rate at basically sitting down at rest. When we're asleep, our breathing rate is about 6 liters per minute. If we're out running around, jogging, playing basketball, our breathing rate might be 14 liters a minute. So 10 liters a minute is average activity. The sampler particles are drawn into the sampler through that intake orifice, and behind that intake orifice is either a drum with a 7-day clock mounted on it for 7 days sampling, and that's what I typically use. There is an alternate head for 24-hour sampling, but I'm just going to briefly describe the 7-day head. So the clock rotates by the intake orifice at 2 millimeters per hour, and mounted on the clock is a drum, and mounted on the drum is a piece of tape, a cellophane tape that's been greased, and it passes by that orifice at 2 millimeters per hour. At the end of 7 days, the drum is removed, the tape is removed from the drum, and it's cut into 24-hour segments. These are mounted on microscope slides and stained and made into permanent slides and then analyzed microscopically. So we can analyze for either an average daily concentration or hourly concentrations. Data from the past 29 years tells me that the top 10 pollen types in the Tulsa atmosphere are oak, ragweed, cedar family, mulberry, elm, sycamore, and I'm not going to read them all. So on a yearly basis, cedar pollen is usually in third position. And depending on the weather, there are times when we see modifications in these. Ragweed, hot, dry summer, ragweed might be low, and Cupressaceae may actually move up into the number 2 position. I don't remember any time when oak was moved out of number 1, at least not in the past 20 years. So if we were to look at pollen seasons and we look at the way allergists define pollen seasons, you will see that they think of trees as pollinating in the spring. And in Tulsa, our tree pollen season starts in early February and it lasts through mid-May. Our highest levels are in April. Of course, though, as a botanist, I know perfectly well that other trees pollinate at other times of the year. Grasses, the peak pollen season for grasses is late May through early June, but of course we also have grasses that are producing pollen all summer and into fall. Weeds, the chief culprit, of course, is ragweed, and then tends to peak at the end of August through October. And again, I want to say that we, of course, have other weeds starting in the spring. We have weeds at low levels right in the midst of the tree pollen season and continuing all summer. But ragweed is the main culprit, and the estimates are that one ragweed plant can release 1 billion pollen grains. And that amounts to something like 1 million tons of ragweed pollen produced each year in North America. And I'm not sure who made this estimate. I've never been able to trace the original source, but that's been in the allergy literature for literally decades, since I first started looking at airborne pollen levels in the early 1980s. So this is a graph that shows the average daily concentration of airborne pollen in Tulsa. My samplers are on 365 days a year. And I picked this year out because it did have a high level in January, and you'll hear soon that this is in fact a juniper or a cedar species. And we have several peaks in January, but typically we see our local tree pollen increasing in February, March, peaking in April, and this peak in May, this shorter peak, this is the height of the grass pollen season. Normally the grass pollen season here has its highest levels the third week of May. And then levels kind of decrease dramatically through much of the summer, increasing in late August with the weed pollen, and this is primarily all ragweed. And we'll talk about other pollen in October and November in just a bit. So I want to define some of the pollen metrics that I'll be mentioning as we go through. Average daily concentration is from our Burkhardt samples, and these are expressed as pollen grains per cubic meter of air. So the pollen we count under the microscope, we determine how much is on the whole slide, and that's divided by the volume of air sampled in 24 hours to give you a concentration of pollen grains per cubic meter of air. Some of the terms that we use include the cumulative season total, and some references refer to this as the pollen index. Start date, end date, and these are not the date that the pollen first appears, but usually it is a percent of the cumulative season total, and it may be when the cumulative season total, when the date when you have reached 1% of the cumulative season total or 5% of the cumulative season total. Other researchers have their own definitions. They may say the first date when a particular pollen grain reaches a concentration of 10 grains per cubic meter or 20 grains per cubic meter. And the end date is usually the comparable date at the other end of the season. So if the start date was 1% of the cumulative season total, the end date would be 99. If it's 5%, the end date would be 95% of the total. And of course, the season length is between the start date and the end date, and the average concentration for the season is usually determined only from the start date and end date. The peak concentration is just that, and of course the date of the peak is self-explanatory. So we're all here because of our interest in Juniperus. So let's switch, and for the rest of the time we'll be talking about that. We have 5 species of Juniperus in Oklahoma. 3 are important allergen types. Eastern red cedar, which pollinates here in February and March primarily. I usually say February through April. Juniperus ashii, or mountain cedar, which is a winter pollinator. Juniperus pinchodii, or red berry juniper, a fall pollinator. So we actually have cedar pollen in the air for 7 months of the year, from October in the fall all the way through April in the spring. There may be various ornamentals that contribute to the spring Cupressaceae pollen levels. And so we actually should just be saying Cupressaceae pollen rather than Juniperus pollen. And I tend to go back and forth using one term or the other. And an important thing to point out is pollen from juniperous species are not distinguishable from each other or from other members of the Cupressaceae. So, and again, reminding you that it's the third most abundant pollen in the Tulsa atmosphere. And this is what the juniperous pollen looks like, and these are stained. Our pollen stain makes pollen a reddish purple color. Without stain, the pollen is colorless. So we do add stain to help us pick out the pollen features. So I'm including another yearly graph, and this is from 2005. And I want to point out 3 high levels of juniperous pollen. The first in January, the peak of 1277 was all juniperous Ashii pollen. In March 4th, even though the level was over 2,000, 1,976 pollen grains were actually Juniperus virginiana. And then in October, we had Juniperus pinchodii with a level of 1,061. Now, what is so remarkable about these 3 peaks is that the only one that is local is that March peak. The Juniperus virginiana is in the Tulsa area. Juniperus ashii, the closest population is in the Arbuckles Mountains, about 200 miles southwest of Tulsa. And Juniperus pinchodii is even further, it's in southwest Oklahoma. So these are all been carried to the Tulsa atmosphere by long-distance transport. So we're all well aware of the encroachment of eastern red cedar across the Great Plains. Oklahoma is no exception. And we have seen the pollen levels increase as well. This is a typical scene in Oklahoma, just west of Tulsa. And I'm sure everyone on this is familiar with scenes just like this. Again, the pollen cones and the pollen. And one of the interesting things about Juniperus virginiana is that the pollen season looks different every year. And that's largely because it pollinates in February and March. And the weather in Oklahoma is just as unpredictable in February and March as it is in Kansas and Missouri. And we could have 70-degree weather one day, and 2 days later we can have 12 inches of snow on the ground. And so we see the daily juniper, and this is only Juniperus virginiana pollen or Cupressaceae pollen, and you can see the graph runs from February actually through the end of May, and you can see the big dips when we have had adverse weather coming in. Even 29 years averaged doesn't smooth it out. For many other pollen types that are pollinating later in the year, when I've averaged 10, 20, 25, 29 years, what we get is really a nice, smooth, almost normal distribution. But we still see the peaks and valleys because of the unpredictable weather in February and early March. So when we look at the cumulative season total since 1987 to including through this spring, we see a definite significant increase in the cumulative season total pollen. And you can see we even have a couple of years with very, very high levels. And these were years when we had really good weather in the spring and the pollen levels were quite high. The yearly peak concentration has also seen a significant increase over time. And When we've looked at meteorological conditions, we have temperature is the main driving factor, and this is not surprising. Rainfall, snow, high relative humidity are all negative factors that influence pollen release and airborne pollen levels. Sunshine, I have that in a lighter color. We don't have all 29 years of meteorological data, so we're still trying to get that complete for the 29 years. But clearly, the driving factor is the temperatures. Wind also has a factor, and these are the wind directions broken down into the 8 cardinal directions, and the pollen levels on those days over the 29 years of Juniperus virginiana pollen season. And we have our highest pollen when the wind is from the southwest. Now, this could just be a reflection of the fact that when we have warm temperatures, we are likely to have winds from the south or southwest, or it could actually be that we're bringing in some Juniperus virginiana pollen from southwest Oklahoma, southern Oklahoma, Texas. More work still needs to be done on that. Some of the confounding factors with Juniperus virginiana is the possible overlap with mountain cedar, and that's in late January to early February. There's also the possibility of long-distance transport of eastern red cedar, Juniperus virginiana, both pre-season and post-season. Warmer January temperatures in Texas and Oklahoma may permit the eastern red cedar pollen season to start in January and possibly co-pollinating with Juniperus ashii. And late in the season, when we get occasional registrations of Juniperus pollen in April and May, this may coincide with northerly winds bringing eastern red cedar, or bringing cedar pollen from regions north of Oklahoma. So let's switch to Juniperus ashii. I've done a lot of work on this. This is an interesting species. It has a limited distribution, largely the Arbuckle Mountains of Oklahoma, Edwards Plateau region of Texas, and scattered areas of the Ozark. It's highly allergenic. It causes what people in Texas call cedar fever, and a large number of patients here are sensitive to cedar fever, or sensitive to mountain cedar pollen. It pollinates in midwinter, and there really is no other pollen in the atmosphere when Juniperus ashii is pollinating. And so pollen that we pick up in December and January, cedar pollen, is likely mountain cedar or Juniperus ashii. And ever since I first started air sampling, we've been recording this in our sampler since 1980. This is the distribution, and there supposedly is a small population in Mayes County in northeast Oklahoma. About 5 years ago, I had a graduate student go out there, and he could only find one tree that he felt certain was mountain cedar. Everything else he was positive was eastern red cedar. The Arbuckle Mountains, and of course the large area of the Edwards Plateau in central Texas. This is the Arbuckle Mountains in southern Oklahoma, a beautiful hillside shot, and this was taken in December, probably 25 or 30 years ago, so everything green is Juniperus ashii. And a close-up of the cones. and a close-up of pollen release, and you'll see another one later on, and the pollen grains. And as I said, we have been picking it up about 48% of the days, not every day, but about 48% of the days we do pick up mountain cedar pollen. And the concentrations are typically low. However, we have picked up very high levels based on the National Allergy Bureau's definition of very high as over 1,500 grains per cubic meter. We forecast this for 17 years and issued forecasts from Texas and also southern Oklahoma. And for a few years, we had a forecast we were issuing in Arkansas, but we did not have any sampling stations in Arkansas, so we discontinued that. So the forecast has two components. A release forecast based on the meteorological conditions. For example, sunny weather, 65 degrees, no rain, it's going to be perfect for pollen release. On the other hand, 32 degrees and the possibility of sleet is going to mean very little pollen release. And then we use trajectories, high split trajectories from the NOAA website to determine where the pollen will travel. And you'll see some more trajectories coming up, and this was just an example of a forecast from 2 years ago, 2.5 years ago, and just indicating, this is from the Oklahoma, the pollen was going to travel to the northeast and then curve into Arkansas. We projected that the exposure risk in Tulsa was high. And of course, the weather was good for pollen release that day, and the trajectory showed us where it was going. So during the 17 years we did the forecasting, we had over 125 days that had moderate to very high levels. Moderate is 15 to 90, high over 90, and very high over 1,500. And there were 5 days when we had very high levels. Now, again, the amazing thing is that these high levels is pollen traveling at a minimum of 200 miles and more likely 500 to 600 miles from central Texas. And how far can it go? So I have a colleague in London, Ontario, and he, for a while, was sampling year-round. He didn't do this every year, but in 1999, he tells me, I have some of your pollen. And on January 27th, he recorded 58 pollen grains per cubic meter. And trajectory showed that the source was the Texas population. And in our forecast for that day, we indicated that the pollen had the potential to travel long distances. There was a frontal system coming in, and the wind speeds were high, and the trajectory showed that over the course of 48 hours, it was traveling vast distances. We have subsequently shown, one of my graduate students is working in the molecular area, and she has shown that the pollen traveling to Canada was indeed Juniperus ashei. Extracting the DNA and sequencing the results, we have proved it. So we're really excited about that. 6 years ago, I started a project with a group of researchers, including scientists from NASA, the University of Arizona, and the University of New Mexico. And this came about from a project that they were doing forecasting dust storms. They had a dispersion model that they called DUST, they called DREAM for Dust Regional Atmospheric Model, and they were using it to track dust storms in Arizona and New Mexico. And cedar pollen is a major problem in Albuquerque, and some of the researchers at the University of New Mexico said, can we adapt this to forecasting cedar pollen? And can we use remote sensing to determine when the cedar trees are ready to release pollen? So this was their plan, and they had everything except someone who knew anything about cedar pollen, and that's how I got involved. So I got a call asking if I wanted to be involved in a proposal they were writing, and it got funded, and so I had a whole group of new colleagues, and plus I spent a lot of time out in the field in Texas and New Mexico. So here's their thinking, that when cedar trees are young or when the cones are not mature, they look green. As the trees mature, they develop, the pollen cones gives the tree a brownish cast. And here, this tree on the right here, shows very high concentration of cones, and so the whole tree has a brownish cast. The people at NASA doing the remote sensing did determine that they could in fact tell the difference in the reflectance data between these trees, between young trees and immature trees, and female trees and male trees with mature pollen cone. So this was the whole premise behind the project. So my group was involved in the air sampling and the field work. So we were sampling in the field in cedar woodlands in New Mexico, Texas, southern Oklahoma, and we were to characterize the juniperus populations in around each sampling location, determine the potential pollen production at each location. There were lab components to determine the pollen grains per cone, and additional studies on the influence of humidity on the weight of the pollen grains. So I see my time is getting a little bit short, so I'm going to move along. There were three components: Juniperus ashii in Oklahoma and Texas, Juniperus pinchodii in Oklahoma and Texas, and in New Mexico, Juniperus monosperma and scopulorum. These were the populations we were studying. You don't need the locations, and this is a map of the ashii locations. Southern Oklahoma, we had two sites within about 5 miles of each other, and then across the Edwards Plateau. And this station was only there during the second year, and that was just south of Dallas. The sites were quite diverse, and this is really best seen here. On the upper right is Sonora, which is very dry. It's out in West Texas, and it's very dry. And lower right is San Marcos, and that's between Austin and San Antonio. And on the left is Balcones National Wildlife Preserve, just northwest of Austin. Trees in Balcones were 20 feet tall, 25 feet tall, and my graduate student and my colleague had to climb the trees to figure out if they were male or female trees. Just some of the summary data from one season. We had some very interesting, and I know I don't have time to go over this all, but I want to point out some of the hourly peaks at Junction. The peak hour at 70,000 pollen grains per cubic meter of air. Some of the pollen cone data, I think, is part of the most interesting part of the study. We were looking at collecting cones from the trees and determining the potential pollen production. So out in the field, we had to examine the trees and we had to assign a value, and primarily it was 1 for a low cone producing tree or 2 for a high cone producing tree. And then we picked 5 low cone producing and 5 high cone producing trees, and we tried to do this across the population area, not all from one place. And then we harvested the vegetation from 1/8 of the tree, and then we took all of that vegetation back to the lab where we counted the cones. And I'm going to skip forward and talk about the cones. And on the low cone producing trees, when we counted 1/8 of the tree and we multiplied through by 8, so the number of cones varied from almost 13,000 to 137,000, and the high cone producing trees from 93,000 to 1.37 million trees. When we estimated the number of pollen grains per cone, we found that the mean value across the population was 400,000 pollen grains per cone. Now that gives you in the realm of from the lowest low cone producing tree to the highest high cone producing tree, the range of 5 to 550 billion pollen grains per tree. So NASA's idea was that we can possibly forecast and develop better dispersion models that will show where the pollen cloud is going over the span of 2 or 3 days. And this was the hoped outcome. Now, we got our part done. Unfortunately, the modelers, the people doing the modeling and the people doing the remote sensing are still trying to put this piece together. So this part is still ongoing, and I think the group is still looking for more funding to continue this. At this point, I know I'm running out of time, and I can stop here or talk a little bit about the last pollen, Juniperus pinchotii. And I know you want to ask questions, so if you would prefer, I can skip to go to the questions.
So let me just encourage you to start typing your questions in, and while you're thinking about those, I think Estelle could probably continue, but at this point, do you want to answer Jamie's question?
Sure. You know, I saw that question and I almost started to enter it in the middle. Most of ours are tree species, and I have to say I'm basing it on Tulsa, and Tulsa is in the part of Oklahoma that we call green country. And we have a lot of trees. We're at the edge of the eastern deciduous forest biome. If you go northeast of Tulsa, if you go northwest of Tulsa, you get into the prairie. And if you go west, we slowly leave the deciduous forest biome and you enter the post oak, blackjack oak habitat, and you still have a lot of oak and cedar pollen there, but not the other pollen types. Those start to drop out. So I think even in Oklahoma City, we see many more non-tree species. So I think Tulsa is reflecting deciduous forest area. So let me just flip through the last few slides on the Pinchotii while you're asking questions. Is that okay?
Yep, sounds good.
All right, so Juniperus pinchotii is typically known as red-berry juniper because the female cones, instead of being blue like we have in eastern red cedar, are red. So a perfect name. They tend to be smaller, scrubbier looking trees, and part of that is the fact that their habitat is really western Oklahoma and West Texas, where there's much less rainfall and literally a lot of desert around them. So we picked up a fair amount of pollen at our pollen sampling sites. I didn't include one of the sites, Junction, because, let me go back. Junction was really on the very edge of the Pinchotii population area. And even when we were in the field, we didn't see that many Pinchotii trees, but we wanted to keep the sampler there. And so this is some of the same data that you saw with the mountain cedar, and again, the peak hour at 67,000, seeing some very high levels. And this is Juniperus pollen in the Tulsa atmosphere during October and November. And you can see the percent of days where we picked up pollen, and generally the concentrations were low, and the peak in all the years was 1,061 back in 2005. So on that day, when we looked at the hourly concentrations, the nice thing about the Burkhardt sampler is that you can keep the slides and go back and reanalyze. And when we reanalyzed them for hourly levels, we saw that we did have some very high levels early in the morning, suggesting it was coming from a distance. And we did backward trajectories looking for where the pollen came from. And the backward trajectories indicated, and I'm sorry, this is the backward trajectory here on the left, and here's Tulsa there, and it indicated that the pollen was coming from West Texas 36 hours earlier. And then we ran forward trajectories to double check, and sure enough, across the Juniperus pinchotii population distribution, this is where the pollen was going on 36 hours ahead of when it landed in Tulsa. But in October 2010, we had other data that we could add because we were sampling out in West Texas. And so the peak in Tulsa in 2010 was on October 25th, and the average was 106, and the hourly peak was 600. So we, again, doing the backward trajectories showed us that it originated in Southwest Texas about 8 hours earlier, and then forward trajectories again from that area showed us where in fact it was coming over Tulsa. And then in Sonora and San Angelo, these were the concentrations that day, and this was when the peak that we got in the afternoon left either or that area sometime that morning. So I think we really proved that source in that case. So the conclusion, Cupressaceae pollen in Tulsa has shown significant increases over 29 years. This coincides nicely with the population increases in Oklahoma as well as elsewhere. and this poses a significant risk to individuals sensitive to cedar pollen. And then both Juniperus ashii, Juniperus pinchodii populations bring us pollen during other times of the year, and these can travel long distances. And so this is Juniperus ashii pollen. This was shot with a telephoto lens from about, I'm not exactly sure how far the person was away, but this was from our sampling site in Junction, from the field assistant who was monitoring our sampler, and he shot this along the hillside. And that is all pollen.
Yeah, Estelle, I see a couple of questions, or I think it's one question.
Okay. Yeah. Has any studies or data collected been for salt cedar? No, I have not. I do think that they, in the region where those trees are going, if there are any people monitoring airborne pollen, they may be, in fact, looking at that, but I have not. So,
I was wondering, so since the pollen does travel so far, is it actually able to fertilize trees at that distance, or might there be anything locally about the DNA that might, the pollen's moving but not actually being productive?
Yeah, I suspect that if there were female mountain cedar trees that far, yes, it could. We did, a few years ago, we did check some of the pollen that we picked up in Tulsa for viability, and they were viable. And this was just a quick fluorescence stain, and viable pollen picks up the fluorescent stain, non-viable does not, and it was picking up the stain, so indicates that at least getting to Tulsa, there would possibly be some cross pollination. So that means that there might in fact be some between the populations in Texas and southern Oklahoma, and maybe even Texas and Arkansas, that there would be some cross-pollination occurring.
The other question that came to my mind, so most of the species that you're really concentrating on are susceptible to fire as a management tool. And what are your thoughts in terms of the air quality trade-offs for using prescribed fire and the smoke produced versus the health effects on the increase in pollen?
Oh boy, that's a tough one. I mean, it's one of those catch-22. Yes, let's cut down, let's have prescribed burns, but if you have asthmatics nearby, they're going to be suffering there as well. That's no easy answer, because of course with the cedar trees, if you don't have prescribed burns and you do have a fire, you could have the same pollutants occurring there as well. There was even there was one study that showed that people who were susceptible, who were allergic to mountain cedar, during a prescribed burn in Texas, there were, I apologize because I don't know if it was a prescribed burn. There were some mountain cedar trees being burned, and the people who were allergic to it were having real great difficulty. So was it the allergen or was it the particulates from the smoke? I'm not sure the paper made that distinction. They just talked about the reactions.
Mm-hmm. All right. Are there any last questions before we wrap up?
I see one more. Once a tree is burned, it's out of the pollen business. Yes, that's certainly true for mountain cedar and eastern red cedar, but apparently Juniperus pinchodii, the red berry cedar, does resprout.
Mm-hmm. That's true. Yep. All right, Estelle, I want to thank you for a very interesting webinar, and I have recorded it, so we'll get that placed onto our YouTube site and our website as soon as possible. Estelle, would you mind typing your contact into the chat box in case anyone would like to
Sure.
contact you in the future?
Absolutely. I'm going to go ahead and just put my email address. Is that okay?
Sounds perfect. All right, and I want to thank all of you for joining us today. And you can find the Great Plains Fire Science Exchange website at www